Welcome back, everyone. So we are going to go into part 2 of topic 4, the genetic blueprint and energy production. So in our last video, we learned about how DNA is structured. So in this video, we're going to learn about how DNA is replicated. So by the end of this video, you should be able to explain the process of semi-conservative DNA replication. Right? So just one learning objective this time. All right, so every time we need to make a new cell in our body, we need to first make a copy of our DNA. Okay, so each one of our cells in our body have a nucleus, and inside the nucleus has our entire genetic code, right, on our DNA. And so prior to cell division, we need to make a copy of that genetic code so that it can pass into the daughter cell. So we need an identical copy of it. So we're going to talk about how that occurs. How can we take our entire genome, or genome is just basically our entire genetic code, and copy it pretty precisely so that we have two copies of it. One can go into each of the daughter cells after replication. So this process is known as semi-conservative replication. And why it's called semi-conservative? Well, semi means like half, right? Conservative, think old. So it's because when we are at the end of replication, we have two copies. One of the copy, so each of the copies, or each of the double helices, on each of the double helices contains one old strand and one new strand. So that way you have like one of the old strands attached, which is the conservative part, and one of the new strands. Okay, and we'll go through that process. Now this entire process occurs in what we call the cell cycle, and we'll go more into the cell cycle later on. I can't remember which topic, but it occurs before we actually go through the cell division, which is the process of mitosis and meiosis. So it occurs in this special phase of the cell cycle called the S phase. And again, we'll go into that later on. You don't need to know about it right this second. Okay? So we call this the semi-conservative replication, where at the end you end up with each strand, each DNA molecule with its double strand. One strand will be old, one strand will be new. Okay. So let's talk about that. Okay, so here is the original strand. Okay, so here is our double helix, and by the end, notice the old will be in gray. Okay, so by the end of DNA replication, you'll have two double helices, but of each double helix, one will have the old and one will have a new. Okay, so here's new and old. So you can see how it's semi-conservative. So each of these DNA molecules are made of one old and one new. So that's why it's considered semi-conservative. So semi, half old, half new. Okay, and this involves basically 3 steps. And this is the basic. Well, we'll go into more detail, but it's basically just 3 steps. The first step involves that double helix unwinding. So remember, our double helix is in a helical structure, and so first of all, we have to kind of unwind it. So it looks more like a ladder instead of a double helix. And then we have to break apart those hydrogen bonds that are holding the complementary base pairs together. Okay, so we have to unzip that. And then we need to add in complementary new bases to the complementary parental strands. Okay, so if we're unzipping it, zip, okay, here we have a parental strand that's kind of free. Here's a parental strand that's free. Then we can use this strand as a template to add new nucleotides to the new strand based on the complementary base pairings of the old. Okay, so if the complementary base pairings, if the parent strand says adenine, well, what binds to adenine? Okay, we have thymine. Okay, so a thymine nucleotide will get added to the new strand. If we have guanine on the parental strand, then a cytosine will get added to the new strand. Okay, same with the other side. If we have a thymine over here, then an adenine will get added here. If we have, cytosine, then a guanine will get added, and so forth. Okay, so each new DNA nucleotide will get put in place based off the parent strands as the template. Okay, and it'll keep going until the entire thing has been copied. And at the end, you get two completely identical double helices. And that's it. Pretty simple. So we're going to watch a video that kind of goes into a little bit more depth. Some of it is going to be a little bit beyond what we need to know specifically for this course. So you don't really need to go too much into detail about like the, too much of the enzymes and things like that, but just kind of pay attention to the process and how everything is working, okay?
DNA replication is defined as the process by which an organism's original DNA is used as a template for the production of a new complementary DNA strand. An enzyme called helicase unwinds the original DNA's double helix, creating a replication fork. Next, an enzyme named DNA polymerase III works down the leading strand and up the lagging strand of the replication fork, synthesizing two new strands of DNA by taking free nucleotides and pairing them with the complementary bases on the original DNA template. The process of DNA replication is described as semi-conservative because the two copies of DNA produced each contain one strand of the original DNA and one entirely new DNA strand.
All right, so that was a pretty good, easy video to just see the process. All right, so let's do our review question. So DNA replicates by which of the following models? Is it, well, none of these. So let's talk about the three possibilities. B, dispersive, C, semi-conservative, or D, conservative. So if you chose semi-conservative, I would agree with you. So DNA replicates by semi-conservative model. So that means by the end, each strand will contain half old and half new. So that is it. So now you should be able to explain the process of semi-conservative DNA replication. Okay. So our next video, we're actually going to switch gears. We're going to learn about energy. So we're going to take a look at how are energy and metabolism related. So we'll see you then.